mRNA targets via HITSCLIP. So first, let's talk about microRNA first. So I think most of you know this already. So, but just to recap, microRNAs are like non-coding RNA that plays a role in like post-transcriptional gene expression, regulation, and RNA silencing. So it was discovered by the Ambrose and Revcon groups in the early 90s. And these are small. These are like average of like 22 base pairs. And one characteristic is it commonly binds with the 3' UTR to induce mRNA degradation. Although this is the common behavior, interactions with the 5' UTR coding sequence and gene promoters were also reported. So how does this work? So first, we have like this primary microRNA that looks like a hairpin. And then the enzyme called dicer like trims this hairpin loop. And what is left is like a double-stranded microRNA duplex molecule. One of the strands will like will like bind with a protein group, which is an Argonaut protein, and then the other one will, which is called the passenger strand, is like discarded. And then this protein microRNA complex will then bind, will then pair up with the messenger RNA, and then afterwards, microRNA protein complexes presence will block the translation as well as speed up the deadenylation, whatever, the degradation of the mRNA. So that's how the process of silencing of the mRNA works from the microRNAs. So here's just like a brief timeline of the developments in the microRNA research. So as I said earlier, it was discovered in the early 90s, 1993. They discovered this Lin4, and they discovered it from like the C. elegans. And yeah, so basically this is just the summaries of the tools and the technology and the experimental technology that are used for microRNA research. So we have mRNA-seq here. So this is like one of the first high throughput methods that uses like just like an RNA-seq for microRNA. But for this one, we'll be focusing on HITSCLIP. So what is HITSCLIP? So HITSCLIP is the high throughput sequencing of RNA by cross-linking immunoprecipitation. So what it is really is it is a genome-wide means of mapping protein RNA binding sites in vivo. So what happens is that so you have a tissue, and then this tissue is like treated with UV irradiation. So the UV irradiation induces like covalent bonds between nearby proteins, so like between proximal proteins and RNA. So and this bond happens only on the sites of direct contact. So and during this process, the UV treatment also introduced some mutations in some of the clip tags at the cross-linking sites. So okay, so once the protein is like binded on the what they call this, on the RNA, it's only a matter of like filtering only those protein of interest that we want to study. So that's where like the immunoprecipitation comes in. So they do the immunoprecipitation to make sure that we only get the proteins that we want. So basically this is the process. So once after the UV irradiation thing, so the cells will be lysed, and from the cells will be lysed, and then they will like get only those they will perform the immunoprecipitation here, and then what they do basically is like after attaching the adapters, they will like digest, they will like perform like a protein digestion, so that there will only be like a small peptide that will remain. And then from here, they will do like a reverse transcription. So to generate the cDNA, and this cDNA will be the one that will undergo the high throughput sequencing. So, and there are other methods. So this is the HITSCLIP that we discussed. So there's also like what you call as the PARCLIP. So this one, I think what happens here is that they add like a photoreactive ribonucleoside on the on the RNA or something. So yeah, I'm not really sure, but something like that. And then there's also this iClip wherein they do this circularization. What happens is that for like HITSCLIP, um they do reverse transcription on the whole thing. But for here, I think they truncate on the site uh of the peptide that remained, and then they do like the circularization thing. So yeah, that's something like there are other methods for like for like for doing ClipSync. So that's just the point here. So HITSCLIP for mRNA. So remember this. So HITSCLIP is actually designed for like looking at the RNA protein interaction. So for this, what we're looking is our target protein is like this Argonaut protein. So this is what we are actually trying to target. So this is what we're using the clip for. So I guess so. Yeah, so we perform clip on the protein Argonaut, and so according to this paper, the characteristic mutation from when the cross-linked RBP is Argonaut are specifically deletions. So this will be important later when we're like processing stuff. So yeah, so how is the workflow now for this analysis? So what I did first is like I got like I got FASTQ files. So of course, if you have FASTQ files, first you want to see how it looks like, and and the FASTQ files I received looks like this. And actually, when if you're looking at this, it kind of looks really good. So I said, okay, it's good. So let's go to the next stage or something. But then when I went to the next stage, which is like the mapping phase, I had some problem wherein it's not really it's not giving me like good results. Like it actually mapped really bad. So when I look into why this is happening, I realized that I did not know that it's really short. So I just realized that later, and I also realized that it's actually like really most of these are just adapters. So it's really just this small area that is the ones that is of interest, and the rest are like adapters. So you have to you have to trim that before you like do do uh do the next step, do the downstream analysis. So okay, so after that, so I just did some mapping. So here are like just some popular mapping softwares. So like we have the star, which is like the star, I think, and then HiSat2. This is what I use. I use HiSat2 because yeah, I tried running star and it's like so heavy with the RAM, and I just failed running it on my computer. So but HiSat2 really did good. So that's what I used. And then afterwards, so now we go to the exciting part, which is the peak calling. So several tools have already been developed to analyze like a single clip dataset, so ClipSync dataset. So we have the Paralyzer, Eclipse, Piranha. So, but these tools never don't really have that much capability for like comparative analysis. For example, if you have like a wild type versus a KO thing, so you knock out like one microRNA and then you compare it with the wild type. So what you can do is there are like some ad hoc methods. So first is you can analyze the conditions separately and use like coordinate overlapping by bed tools to obtain common and differential binding sites. But this method will not detect differential binding site intensities for two regions that are significantly enriched, and there is also like it's also oversensitive to like cutoffs. So what I did is one of the recommendations I saw is other than that, there's also like challenges for like a ClipSync versus like ChipSync is a method for like inspecting DNA um, protein interaction, and ClipSync is for like RNA protein. So again, so the problem that was stated is that it's strand specific. So, and then there are like characteristic mutations that is like kind of important to look at. And then there's also the problem of the short reads, and it also like requires a much higher resolution, like in a matter of like single base pair or something. So here's one of the recommendations that I saw in most papers. So they mentioned DCLIP, which actually has like a comparative, which actually does this differential comparison between like two 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 what you call it, two treatment or two categories. So it is a two-stage approach minus the pre-processing. So for the pre-processing, what happens is that first it collapses duplicate reads into like unique tags, and then it notifies it looks at the characteristic mutations collected on all of these tags. You can actually like set in the input what characteristic mutations you wanna look at. So remember earlier I mentioned that the characteristic mutation for this kind of for like Argonaut is deletion. So that's what I put when I was like processing when I was like creating the script for like rather the run for like DCLIP. And then afterwards, the clusters are divided into bins of small length. So the default is like five base pairs. And what happens is that DCLIP basically compares the tag intensities for each bins on the wild type versus the first condition and the second condition. So this is just the process. So after the pre-processing, so this is the two-step thing that they're doing. So you're just doing like your MA plot and normalization, and then like a hidden Markov model to like set like the states. So here is like the DCLIP output. Here's the state actually for the hidden Markov model. So you have like a 0 for bin with like binding more for condition 2 than condition 1. So, and 1 if it's actually weird. So 1 if it is like bin with like equal binding, so there's no difference between it. And then 2 if it is more on 1 than condition 2. So those are like the 3 states that they have. And so yeah, so this is basically the output. So we have like the ID. So this is the ID of the cluster, and then the chromosome, and then what strand this is, what strand it is, whether it's a positive or negative strand, and then the position. And you can see it's like each of this is like a bin. So this is from 0 to 4, then 5 to 9, etc. So it's like 5 base pair each, and then the state. So which is I explained earlier, and then this is like the probability coming from the hidden Markov model. And then the differential one is basically just on the difference between the differential difference between the normalized tag intensity between like two conditions. And these are just like the information regarding the tag intensities and the mutations of the two conditions. And then the other output is they also have like this D-summary bed. So this is basically just a bed file wherein we have like the chromosome, and then the location start and end, and then this is like the ID, like the what do you call it, cluster ID, and then this one, this is like the average binding strength. So the 0 if the state is equal to 1, which you recall earlier is like it's just an equal state, which means that there's no difference between the two conditions, and then yeah, so and then it's not 0 if there's a differential difference, if there's a difference. And this is actually what I used and what the authors recommend to like use for like ranking genes or ranking the targets. So basically here is my strategy for how I did it. So first I just need to find the clusters that are differentially enriched in the wild types. Of course, if it is knockout, so it's not there, or rather it's weak or something. So and then we can use this coordinate and state information in the DCLIP output. So remember, we have like the DCLIP output here, and it is the state. So we just get like, for example, state 0, and then just get everything in there, and then just get like the information on the position from the position or something, and you create like a bed file in there, and then just make what do you call this, like a bed file, and then just do a, like an intersection. And then once we get the W type only cluster, so for example, this is the W type cluster, we want to further limit so that it's not too many. So we want to further limit our search by including only those that are enriched on the 3' UTR. So, and then we rank these clusters using the average binding strength in the Eclipse summary bed. So yeah, so what we do is just we just do like a series of intersections using like different bed files that I download. So maybe I just show you the notebook that I generated. So here it is. So so here's the decrypt output text. So what I did first is like I just extracted all the wild type clusters. So this so we want to extract clusters that have more binding in condition 2 than condition 1. So this means that the state should be 0. So I just did some filter, and then what did I do? So from this I like generated like a bed file, and then created I think I just created like a new bed file for like the whole type that with the with the wild type whole genes only. So yeah. And then I downloaded this information on the locations of the 3' UTR from the mouse. So this the organism here is the mouse. So I downloaded this information, and I just did like an intersection between like this and our like wild type only the wild type only whole genes from earlier. So 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 what should remain are like the cluster IDs of those that that are differentially that are more differentially binding on the wild type condition, and those also those that are that exhibit like this property on the 3' UTR, right? So and then yeah, so what I did afterwards, so once I got that, I just ranked it based on like the column 4, which is, if you remember, which is the average binding strength. So and then basically that's how I got the gene names. And yeah, so I just took the top 500 genes, and then yeah, what else? And then from there, I just performed like a enrichment analysis on the enricher. But yeah, that's basically how I got like top 500 candidates from this HITSCLIP data set that I have. So yeah, is that clear? Do you have any questions? I think.
How did you get genes all of a sudden? You were
Oh,
dealing with like these base pairs.
I guess so all right, so I downloaded two files from USCSC. So first I got the 3 UTR ref6. So this is like the location of the 3' UTR, and then I also get like the names of the whole this MM10 whole gene ref6 dot bed.
Oh, I
So
see
this
the
is like
reference gene.
information. Yeah. So that's what I did. So I just did like intersections between this tree. So so the intersection between my wild type and then the 3' UTR, and then this this gene annotation. So that's how I got like this. Actually, it's a transcript name, but I converted the transcript name to gene names, and I took like the top 500 genes.